Hello, everyone. My name is Formosa. I'm a lecturer and researcher at the University of Malta. And today I will be discussing two family studies with early onset osteoporosis and the journey we took from high throughput sequencing to the functional testing to validate our findings. So the first family was a 3-generation pedigree of 29 relatives, and their ages ranged from 21 to 81 years. This particular family lived in the southern part of Malta. The proband was a 54-year-old female. She had osteoporosis at the hip, and she was identified during a routine bone density scan within Malta's main general hospital. At the time of her recruitment, she reported that both her mother and her younger sister were recovering from a non-traumatic hip fracture. Her younger sibling was a 49-year-old perimenopausal woman. She had osteopenia and she had already sustained a previous hip fracture. So this gave us an indication that there might be a strong genetic component leading to osteoporosis and fractures in this particular family. And as a result, we sought to recruit as many relatives as possible. So the recruited relatives were subjected to a bone density measurement of the lumbar spine and hip, which was performed on a Norland densitometer. We also collected peripheral blood samples for DNA extraction and also biochemical testing, including a complete blood count, antigliadin test, hormone profile, bone biochemical markers, glucose measurements, and these were done to exclude the possibility of secondary osteoporosis. We also collected demographic and lifestyle factors, as well as a thorough medical history and medication use. Where possible, medical records were reviewed, and we saw that none of the relatives had features of osteogenesis imperfecta or any other extraskeletal abnormalities. Osteoporosis was defined using a T-score of -2.5 or less at the lumbar spine or total hip in postmenopausal women and men over the age of 50, whereas in the case of premenopausal women and relatives younger than 50, we used the Z-score with a cutoff of -2. An affected status, as you can see, is represented by a half or a full black symbol, and this signifies the presence of osteoporosis or a history of a fragility fracture. The lowest T-score or Z-score at the lumbar spine or total hip and the type of fracture sustained can be seen in this pedigree. Most of the affected relatives from this family had osteoporosis at the hip, including the proband, and the youngest individual, a 21-year-old male who happened to be the son of the perimenopausal female who had a history of 2 hip fractures. So we sought to investigate this family by first doing a genome-wide linkage scan, and we selected 22 individuals. We genotyped them on the Illumina human cytosnip12 chip, and we analyzed the data by Allegro using the Easy Linkage Plus software. So we performed parametric multi-point linkage, and we obtained 2 linkage peaks with a lot score of 2.1, and these mapped to chromosomes 12 and 19. We then further scrutinized these linkage regions using whole exome sequencing, which we performed on the HiSeq 2000 Illumina sequencing platform. We used an in-house bioinformatics pipeline to process the raw sequencing reads, which eventually gave us a variant call file, and we applied stepwise variant filtering to narrow down the list of variants in the linkage regions, including filtering based on zygosity in affected individuals following a dominant inheritance pattern. We removed variants with a minor allele frequency of more than 1%, and we removed synonymous and other benign variants. And this left us with 1 variant, 1 intronic variant, and the zinc finger protein 384 gene, which we confirmed by Sanger sequencing in all affected relatives, and it was absent in unaffected ones. So I guess you are wondering why we focused our attention on this intronic variant, since it might not carry so much value, and furthermore, it was already known. However, nothing was known about this particular variant. But let's go a step back. What did we know about the gene at that point in time in our study? So we knew that the gene codes for a transcription factor. It has several cis2-his2 zinc fingers, and these are important for transcriptional activation of several extracellular matrix genes, including MMP1, MMP3, MMP7, and also COL1A1. There was a knockout mouse model that was already published, and it showed that knockout mice had increased bone volume and bone formation rate compared to wild-type mice. These knockout mice also had an upregulation of alkaline phosphatase, osterix, and type 1 collagen, and that ALP upregulation was enhanced via BMP2 signaling. So later on, in 2019, another study was published on this particular gene, and it showed that loss of ZNF384, also known as NMP4, was related to enhanced bone quality. In the case of the variant, it's an intronic variant, as mentioned. It's a single nucleotide substitution, and it is located 32 bases upstream from the intron-exon junction. So for that particular reason, we used different in silico modeling to see whether the variant might be affecting splicing. And indeed, it was predicted that the variant abolishes an exon splicing enhancer motif and the branch site. According to NOMAD and other population allele-based frequency databases, the variant is rare. It is present in less than 1% of some European populations, whereas it is monomorphic in several others. And on that note, we used the Malta osteoporotic fracture study to determine if the variant might be affecting bone mass and fracture risk at the population level in Malta. So the Malta osteoporotic fracture study is a case control collection of more than 1,000 postmenopausal women recruited between 2010 and 2012. Women with a history of at least one low trauma fracture were classified as cases, and most of these cases were fresh trauma fracture cases recruited within 18 hours of fracture. Controls, on the other hand, were women without a fracture history. We performed genotyping using real-time PCR, and the results further strengthened the association with BMD and fracture risk. So as you can see, women carrying one copy of the alternative allele had a twofold increased risk of osteoporosis at the femoral neck, all type of low trauma fracture risk, and also hip fractures. And as you recall, this is in line with the phenotype observed in affected relatives from this family. Moreover, based on the link between ZNF384 and COL1A1, and also from a study that was published in 2009, which saw that variants within COL1A1 can interact with ZNF384, we had genotyping data for a promoter variant in COL1A1, and we found that women carrying the heterozygous variant for both the ZNF384 and the COL1A1 variant had a threefold increased risk of low femoral neck BMD when analyzing these gene-gene interactions. So next, we investigated the predicted effect of the variant on pre-messenger RNA splicing, and we used the PSPL3 exon trapping vector transfected in SAOS2 and HFO B cells. Results from our in vitro work showed that in the presence of the alternative A allele, exon 8 and part of intron 8 were retained, and these are spliced out in the presence of the wild-type G allele. If you look at GTEx data, only one ZNF384 transcript harbors exon 8, and in most tissue, this transcript is very, very weakly expressed. And we also confirmed this in some of our preliminary testing that we performed in bone tissue, it's bone from tibia, and the human fetal osteoblast. We also see that this transcript is very weakly expressed. So in summary, the study confirmed the role of ZNF384 in bone mass determination. The variant, although it is known and intronic, it has been shown to be associated with hip BMD and fracture risk in our 3-generation family, and this was replicated at the population level in Malta. And the variant also seems to interact with the COL1A1 promoter variant. In vitro, the variant affects splicing with exon 8 being retained, and it is important to note that the neighboring exons of exon 8, they code for the cis2 his2 zinc fingers that are important for transcriptional activation. So it would be very interesting to see how this retained exon might be affecting this transcriptional activity of the resulting protein. Of course, keeping in mind the deleterious effect that the gene has on bone mass from the knockout mice models that we discussed before. Our second family was a two-generation family consisting of 12 relatives, including 7 siblings, and this family resided in the central part of Malta. So where possible, again, we performed biochemical investigations, we reviewed medical records, and we excluded any other bone diseases or disorders affecting bone. We performed DEXA, the lumbar spine and hip, on a hologic densitometer, and the same phenotype definition that we described before was applied. In this case, the proband, as you can see here, had suffered a non-traumatic vertebral fracture, and she required the insertion of metal screws along her vertebral column for fusion and stabilization. Unfortunately, no BMD scans were performed prior to her fracture, so we did not really have any BMD results of her lumbar spine. However, looking at the BMD of the total hip, it revealed the presence of osteoporosis. And her daughter, at the age of 43, was already diagnosed with osteoporosis at the lumbar spine and hip. And in this particular family, all affected relatives had either osteoporosis at the lumbar spine or had sustained a vertebral fracture. So we performed whole exome sequencing, again on the HiSeq 2000 Illumina sequencing platform. We applied sequential variant filtering to narrow down the extensive list of variants, and we focused our analysis on the protein impacting ones, including those that were predicted to be deleterious by most in silico tools. And this left us with one nonsense variant in the kinesin family member 26B gene. And again, we confirmed the variant by Sanger sequencing in all affected relatives, and it was absent in unaffected ones. So some information about the variant. So the variant is located in exon 3. It is in the N-terminal of the protein, which is upstream of the kinesin motor domain, and this domain is used for the transport of cargo along microtubules. The variant is absent in all databases reporting population-based allele frequency, and according to the American College of Medical Genetics and Genomics, it is predicted, or at least it is classified to be pathogenic, and it has a score of 5. And the variant, as you can see, is also very highly conserved. According to in silico prediction, the C2T change results in a premature stop codon, and it targets the messenger RNA for nonsense mediated decay. And using Western blotting in post-7 cells, which do not endogenously express the protein, we confirmed that in the presence of the alternative T allele, there is complete protein ablation. Again, we sought to genotype the variant in the Malta osteoporotic fracture study using competitive allele-specific PCR, and we identified 2 unrelated women carrying the variant in heterozygosity, and both of these women had a low lumbar spine BMD, and this replicated what we see in affected relatives from the family. The only issue was that for the second individual, the BMD measurements that we had were obtained after a few years of bisphosphonate therapy, and she had remained relatively stable over the next 3 to 5 years after starting therapy. Unfortunately, we did not have any measurements prior to bisphosphonate therapy or after 5 years after her treatment. So we do not know whether there were any changes in her BMD measurement or if she had sustained a fracture for that matter. Of course, our next step was to assess gene function in relation to bone physiology. At the time of the study, we knew that the gene was expressed in limbs and in the musculoskeletal system. We knew that a complete knockout was lethal in mice, and heterozygotes had severe kidney malformation, and they also had problems with lipid metabolism. However, we did not have much to go on with regards to the role of the gene in bone physiology. So for that reason, we used zebrafish, and the only downside being that the gene is duplicated, so we had to create a double knockout using the CRISPR-Cas technique. So looking at some of the results that we obtained in zebrafish, so the combined alizarin red alcine blue staining revealed that the KIF26B double knockout fish exhibited reduced mineralization of the cranial bones and vertebra compared to the wild type SIBs at the larval stage at 8 days post-fertilization. And they also had reduced alkaline phosphatase at the larva stage. This reduced ossification persisted at the juvenile stage and the double knockouts, and they had a striking reduction in mineralization of the cranial bones, vertebra, the ribs, and their fin rays compared to the wild type fish. And this difference was statistically significant. We also performed microcomputed tomography on 3-month-old fish, and again, it revealed diminished bone mass at the skull and vertebra and double knockout fish. And they also had a reduced BMD and reduced bone volume tissue. So despite the impaired ossification, the overall length and growth of the double knockouts was not reduced, and it was not lagging behind. And this indicated that there was no general developmental delay in these fish. And as you can see, there were no skeletal deformities either at any stage of these fish, of the larvae, juvenile, or adult fish. We also used fish scales in our study. So these elasmoid scales, they cover the entire body of fish. They are composed of calcified dermal bone in a plywood orientation, and they are embedded in the fish's scale. So the nice thing about this scale, these scales is that besides being abundantly present in number, they are easily accessible, they regenerate throughout life, and they harbor the same bone cells, molecular signaling pathways, and mineral content. And the type of calcium mineral that is found in scales is in the form of hydroxyapatite. So the first formed scale is known as an ontogenetic scale, and it develops during ontogeny, and it has these characteristic concentric mineralized ridges known as circuli and a radial network of unmineralized furrows known as radii. When a scale is damaged or lost, the regenerating scale is produced and it becomes visible within 3 to 5 days after removal. And this development is highly conserved, and it takes about 21 days to become fully mature. So for the scale assay, we used 4-month-old fish. We recovered ontogenetic scales from the second or third row and third row above the lateral line from the left flank. And then we sampled again after 6 days to get the regenerating scales. Whereas from the right flank, we removed the ontogenetic scales, and then we sampled again after 14 days to get the regenerating scales. So in that case, we had ontogenetic scales and regenerating scales at 6-day and 14-day time points. And all of the scales that were recovered were subjected to von Costa and trap staining. So looking at the von Costa stain, which demonstrates the presence of calcium minerals and the scale, we see that at all time points, in ontogenic and 14-day regenerating scales, there was an increased demineralized area indicating a pronounced osteoclast activity. And besides this increased demineralization, the scales also had several malformations as indicated by these arrowheads. In the case of trap staining, we saw a difference at all time points. And as you can see, the scales retrieved from the double knockout fish, including the ontogenetic and the regenerating at 6-day and 14-day time points, had an increased surface area of trap positive staining. And this became more intense over time. And again, there were these areas of severe malformation as indicated by the arrowheads. So in summary, we show that this novel nonsense variant is a likely cause of autosomal dominant osteoporosis in a 2-generation family, and also a low trauma fracture history. So the variant was also detected in a case control setting, and we identified 2 unrelated women carrying the variant in heterozygosity, and they also exhibited low lumbar spine BMD, which replicates the findings in affected individuals from the family. We also provide functional evidence in vitro, which demonstrates that the alternative allele results in complete protein ablation, whereas in vivo, our zebrafish analysis highlighted the involvement of the gene in bone mass regulation and mineralization, and possibly creating a target for osteoporosis treatment. And this is precisely where we are at in our studies. So thanks to some recently funded projects, we are currently testing several small molecules that we know are involved in the pathway in which the KIF26B is involved, in the planar cell polarity pathway. And we are also interested to know more about the microRNA and the metabolome of all the families that we have recruited and analyzed thus far. So stay tuned for more findings later on. And finally, our research would not be possible if not for our many sources of funding and our collaborators who have been immensely helpful, supportive, and patient with me over time. Thank you.